Hello everyone, hope you all are doing well. So today we are going to do top 39 of paper 2. Here we will be discussing DNA fingerprinting, and we will also discuss its usage and its role in seed production or seed variety protection. So what is DNA fingerprinting? DNA fingerprinting. So as we all know that we have different fingerprints, so like that we also have different nucleotide sequence in our DNA. A single nucleotide sequence of DNA can never be matching to another nucleotide sequence. So through that we can differentiate everything. So we can define DNA fingerprinting that it is a technique in which an individual DNA is analyzed to reveal the pattern of repetition of particular nucleotide sequence. To reveal pattern of repetition of particular nucleotide sequence So these nucleotide sequence is different for all the animals, plants, human beings. All the species have contained this different nucleotide sequence throughout the genome. So here the unique pattern of DNA sequence is identified by Southern blot technique. This is identified by Southern blot technique. It has been used in criminal investigation and plant variety protection. It has been highly used in criminal investigation and also in plant variety protection, PVP, plant variety protection. And this technique of DNA fingerprint relies on development from recombinant DNA technology, recombinant DNA technique, and allows an examination of each individual's having a unique blueprint, and it allows to the study of every individual's unique blueprint of DNA. Every individual has a unique blueprint of DNA or unique nucleotide sequence. So what is the importance of DNA fingerprinting? You can understand from these points. It is helpful in forensic application, in forensics. It is useful in paternity determination because the nucleotide sequence we have are similar to, not same but similar to our parents. So from that we can determine the paternity. You have seen in many movies or in many serials or that they used to check DNA to determine who the father is, who the mother is. So that is paternity determination, and also for varietal protection. So this was the importance of DNA fingerprinting. Now moving on, what is the procedure of DNA fingerprinting? How do we analyze this DNA fingerprinting? Procedure of DNA fingerprinting. First of all, we will take the DNA sample. Then we extract DNA from that sample. Then we will digest the DNA by restriction endonuclease enzyme, digestion of DNA. Digestion of DNA by restriction endonuclease enzyme. Then we will do gel electrophoresis for DNA probe separation. Gel electrophoresis for DNA probe separation. Then we will do southern blotting technique to transfer fragments through nylon fragments. Then we will use southern blotting technique to transfer fragments, to transfer DNA fragments to a nylon fragment, Then we will do autoradiography to obtain DNA bands. Autoradiography. This is done to obtain DNA bands. So after the bands are made, we will visualize the DNA fragments or DNA bands and mark the difference or mark the similarities. Development and visualization of DNA fragments. So this was the procedure of DNA fingerprinting. Now moving on. Role of DNA fingerprinting in plant variety registration or protection. Role of DNA fingerprinting in plant variety protection or plant variety registration. So first of all, it is very useful to protect plant breeders' right to protect plant breeders' right. And we should know that the old technique has limitations, like the old technique before DNA fingerprint has limitations, like they had they can be influenced by environmental. They had limitations such as environmental influence. Due to changes in environment, their DNA could be changed like this. There was pleiotropic effects. So these were the limitations in old technology, which is not present in DNA fingerprinting. DNA fingerprinting shows high precision that leads us to accurately know what the variety is, what is the breeder claiming the variety is of his or not. So it is high precision. It shows with 100% guarantee. That is why it has high potential to protect IPR of plant breeder. But also we need to establish high laboratory standards, procedure and network all over the world. To make it successful, we need to establish high laboratory standards, procedures and network all over the world, because many a times there are differences between breeders when there is export import. Suppose you are exporting some variety to Australia, let it be, but the breeder of Australia starts claiming that the variety which is exported is been made by him or her. So we need to have a network of labs all over the world, or global network of labs, so that will also enhance our role of DNA fingerprinting in plant variety protection and registration. It has also a high degree of non-tissue specific polymorphism, high degree of non-tissue specific and it has simple inheritance pattern. Which doesn't complicate things, it has simple inheritance pattern. So this was the role of DNA fingerprinting in plant variety protection or registration. Now, what is the role of DNA fingerprinting for identification of cultivars? Role of DNA fingerprinting in identification of cultivars. So we all know that DNA fingerprinting can be called a genetic photograph. Which genetic photograph, and it is not only for individual but for plant, animal, person, anyone. Any living organism has this genetic photograph. So how will we identify cultivars? Using this photograph. So DNA fingerprinting makes it possible for researchers to pinpoint a specific fingerprint. So due to its high accuracy, we can pinpoint to a specific fingerprint and accurately identify seed varieties. Pinpoint specific varieties, that is, we can pinpoint, know which variety is this, which so that is how it is helpful in cultivars, because sometimes there are similar cultivars, similar varieties of a cultivars. So using all techniques, it sometimes become difficult to know which variety is of which cultivar, or which variety is of that cultivar or not, or these varieties of those cultivars are different or same. But in DNA fingerprinting, we can know it pinpointly. SSR markers, I have taught marker based technology or marker assisted selection in detail in previous lectures, so you must see it, so you will know what markers are in this lecture. SSR markers are developed in respective crops, varieties released in future. So we can develop SSR markers in respective crop varieties which we are going to release in future. And markers differentiate all released varieties. Markers are different for all varieties, hence they can differentiate all released varieties. We need to have use of labs for identifying, and it is also useful for in identification related to IPR, that is intellectual property rights. So it can also solve problems related to IPR. So that is all for today. I hope you all understand what the DNA fingerprinting is and how it can be helpful in seed protection or variety protection or identification of cultivations. So for any queries, to comment. If you like it, please press the like button. Do share and subscribe. Keep making notes. That's all for today. Have a nice day. Thank you.